Hello students, welcome to Rayat Online School Education Project, that is the ROSE project conducted by Rayat Shikshan Sanstha Satara, Sadhguru Gadge Maharaj College Karad, conducted here 12th ROSE project 2021 to 2022. Myself, Miss Mohite PS. My subject is biology, and here my topic name is enhancement of food production. Enhancement of food production, that is enhancement means increase in the quantity with the quality of a food production by using biological method. In this topic we see first point improvement in a food production, second point is a plant breeding, third point is a tissue culture, fourth point is a single cell protein, that is the SCP. Fifth point is a biofortification. Sixth point is a animal husbandry. Seventh point is a microbes in a human welfare. Eighth point is a role of microbes in a industrial production. Ninth point is a microbes in a sewage treatment. 10th point is a microbes in a energy generation. 11th point is a role of microbes as a biocontrol agents. And last point is a role of microbes as a biofertilizers. See here, first point, that is the improvement in food production. Food is the essential for all living beings. We know that, but food, it is the biological definition of food is it is an organic, energy-rich, non-poisonous, edible, and nourishing substance that gives us energy and keep us alive, strong, and healthy. We know that green plants prepare their own food through photosynthesis, but animals cannot prepare their food and hence are dependent on plants. Here, food production needs to be improved to meet the increasing demand for food both in terms of quantity and quality. Plant breeding and animal breeding help us to increase the food production. Here first we see plant breeding. Plant breeding is the applied branch of the botany. Here definition of plant breeding, that is the improvement or purposeful manipulation in the heredity of crops and the production of new superior varieties of existing crop plants constitute is called plant breeding. The plant breeding is done to increase crop yield, improve quality, increase tolerance to environmental stresses, make the plants resistant to pathogens, and increase tolerance to insect pest. It is the plant breeding is applied principle of genetics, taxonomy, physiology, pathology, agriculture, and rDNA techniques, etc. to increase crop yield, improve quality, increase tolerance to environmental stresses, and increase diseases and pest resistance. Here, Green Revolution, we know that Green Revolution was the result of a sequence of scientific breakthrough and developmental activities that successfully fought hunger by increased food production. Here, Green Revolution is the scientific breakthrough and developmental activities lead to increase food production with high yields and disease resistance varieties in the wheat, rice, maize, etc. See next slide. In this slide we see hybridization and its techniques. Here, hybridization is the technique of combining together and desirable characters of two or more varieties. It is the chief method that offers greater possibilities in the crop improvement than other methods. By this method, one can create new genetic combinations of already existing characters and new genetic variation. Here we see it produces new genetic combinations and new genetic variations. Here a wide or a distant across distantly related parental plants. The main step of the plant breeding program, that is the hybridization, are as follows. First step is a collection of variability. In this step, wild species and relatives of the cultivated species having desired traits should be collected and preserved. Here, germplasm collection is the entire collection with all the diverse alleles for the all genes in the given crop. The germplasm is conserved by in situ conservation. It can be done with the help of a forest and natural reserves. And ex situ conservation, it is done through botanical gardens, seed banks, etc. Here next step of hybridization is a evaluation and selection of parents. The collected germplasm is evaluated to identify plant with desirable characters. It is an important and essential step. The selected parents must be healthy, vigorous, and should show desirable but complementary features. It is made sure that only pure lines are selected, multiplied, and used in the hybridization. Here in this step, healthy, vigorous plant with contrasting characters are selected as parents and they are selfed to induce homozygosity, that is the pure line. Pure lines means what? The selfed to induce homozygosity, that is pure lines. Next step is a hybridization. In hybridization, the pollen grains from anther of a male parent are collected and then artificially dusted over stigmas of emasculated flowers of a female parent. The seed thus obtained represents the hybrid generation. In the hybridization, here, selection of parent plants with different qualities, then collection of pollen grains from the flower of male parents and artificial cross-pollination by using pollen grains collected from the male parent, and in the hybridization, bagging tagging of the emasculated flower of female parent. The hybrid F1 progeny is selected and evaluated for the desired combination of characters. Here, development of fruit and seed representing in a F1 hybrid generation. Next step is a selection and testing of a superior recombinant. In this step, the F1 hybrid plant showing superiority over both the parent and having high hybrid vigor are selected. Here in this step, selfed to obtain homozygous for the said desirable characters. Next step is a testing, release and commercialization of new cultivars. Here, the newly selected lines are evaluated for the productivity and other features like disease resistance, pest resistance, quality, etc. Initially, these plants are grown under controlled conditions of water, fertilizers, etc., and their performance is recorded. Many high yielding hybrid varieties of rice, wheat, sugarcane, millets developed through hybridization have helped farmer community to attain record agricultural production in India since 1961. This is called a Green Revolution. Here we see steps of hybridization techniques. Here we see chart of steps of hybridization technique. In this chart, first step is a collection of variability from germplasm and gene bank. Second step is evaluation and selection of parents. Third step is selection of parent plants with different qualities. Fourth step is selected parents selfed for 3 to 4 generations to make them homozygous or true breeding. Identification, next step is identification of parents as a male parent, that is the donor. Male parent is a donor and a female parent, that is the recurrent. Next step is a collection of pollen grains from the flowers of a male parent. Next step is removal of stamens from the flowers of the female parent, that is the emasculation, removal of stamens from the flower of the female parent is called a emasculation. Next step is a artificial cross pollination by using pollen grains collected from male parents. Next step is a bagging, tagging of the emasculated flower of a female parent. Another step is a development of a fruit and a seed representing F1 hybrid generation. In a F1 hybrid generation, we see here development of fruit and seeds. Next step is a selection and testing of a F1 hybrid for a combination of desirable characters. And another step is field trials for yield, that is the productivity. And then last step is a testing and the release of a variety. Here we see some Indian hybrid crops. That is the first is a wheat and a rice. In 1960s, wheat and rice production increased. Sonalika and Kalyan Sona are two of the hybrid wheat varieties grown in India. Sugarcane, next is a sugarcane. Saccharum barberry is a native of North India and S. officinarum belongs to South India. They were crossed to obtain varieties such as CO4, 419, 421, 453 at Coimbatore. Next is Indian hybrid crop is a millet. Millet is a hybrid maize, that is the Ganga 3, Jowar, CO12 and Bajra, Nifad have been successfully developed in India. These varieties are high yielding and resistant to water stress. Here we see in the next slide plant breeding for some disease resistance. Plant breeding for disease resistance. Some of the diseases caused in plants are, here we see a table, that is in this table pathogens and plant diseases we can see. First pathogen, that is the fungi. Fungi is responsible for brown rust of wheat, red rot of sugarcane and late blight of potato. Second pathogen is a bacteria which are responsible for black rot of crucifers. And third pathogen is a viruses which are responsible for tobacco mosaic virus. The basic objective of breeding for disease resistance is to develop inherent quality in the plant to prevent the pathogen from causing the disease. Such varieties of plants are called disease resistance plant. The disease resistance plants means plants that prevent the pathogen from causing the disease. Here some disease resistance plants developed are, here we see crops, their variety and resistance to disease. Here first crop is a wheat. Its variety is a Himagiri and its resistance to leaf and stripe rust hill blunt. Second crop is a Brassica. Their variety is a Pusa Swarnam and resistance to disease white rust. Third crop is a cauliflower. Their variety name is a Pusa Subra and it resistance to disease black rot and curl blight black rot. Fourth crop is a chili. Their variety is a Pusa Sadabahar and resistance to disease chili mosaic virus, tobacco mosaic virus and leaf curl. Next point, that is the mutation breeding. Mutation is a sudden heritable change in the genotype caused naturally. Natural physical mutagens are high temperature, high concentration of CO2, X-rays, UV rays. They can be induced by agents called mutagens. आता here mutation, they can be induced by agent called mutagen. Here we see chemical mutagens are nitrous acid, EMS, that is ethyl methyl sulfonate, mustard gas, colchicine, etc. Mutagens cause gene mutation and chromosomal aberration. The treated seedlings are then screened for resistance to disease or pest, high yield, etc. Here give some example, that is NPA36 variety resistant, Indore 2 variety of cotton that is resistant to bollworm, Regina 2nd of wheat, Rust variety of cabbage resistant to bacterial rot. Next point is plant breeding for developing a resistance to insect pest. Insect being herbivorous incur heavy loss in the quantity and quality of crops. Resistance in a crop can be developed by following ways, that is the development of morphological characters, that is like hairy leaves in cotton, and wheat develop vector resistance from jacid and cereal leaf beetle respectively. Solid stem in wheat lead to resistance to stem borer. Biochemical characters provide resistance to insects and pests. For example, the high aspartic acid and low nitrogen and sugar content in maize lead to resistance against stem borer. The nectarless cotton having smooth leaves develop resistance against bollworms. Some pest resistant varieties are, here we see some pest resistant varieties, here crop name, here variety name, and here insect pest. First crop is a brassica, their variety is a Pusa Gaurav, and insect pest is a aphids. Second crop is a flat bean, here varieties are Pusa Sem 2, Pusa Sem 3. Insect pests are jacid, aphids, and fruit borer. Third crop is okara. Their varieties are Pusa Sauni, Pusa A4. Insect pests are shoot and fruit borer. Here our first lecture is finished. Thank you.